In the last lesson, then, we introduced ourselves to two basic kinds of cells on planet Earth. We have these small, simple bacteria-like cells, and then large complex cells like we find in animals and plants, and the single-celled versions, amoeba and euglena. In the rest of this lesson, we'll continue to look at some similarities and differences. Now, all cells must get building materials and energy from their environment. For bacteria, they're the smallest, simplest kind of cell. They are literally importing molecules. So that's what they eat, molecules. And you'll recall, the cell is alive, but of course the food that bacteria are eating, these would be non-living molecules. In the case of larger cells like an amoeba, it's a predatory cell, so it gets its building materials and energy by eating other living cells. So big cell eating the little cell. In fact, the paramecium eats bacteria. So paramecium are bigger than bacteria. Paramecium eat bacteria for lunch. So cells are bundles of useful molecules and therefore a source of energy and building materials. So it makes sense that in the history of life, cells evolved the ability to extract useful materials from other living organisms, and so we get predator-prey relationships. Let's take a look at a scene on a microscope slide of an amoeba eating a paramecium. Now in this scene we have the amoeba here, the dark kind of silver thing here, this big blob, that's a one-celled predator, and then its prey is a paramecium. So here we see the amoeba extending pseudopods, the goal of which would be to completely surround the paramecium to form a food vacuole. Now here we see two food vacuoles recently formed, and we have living paramecia trapped in these chambers. So these are digestive chambers. The amoeba is going to turn a living paramecium into a bundle of useful nutrients. Here we're peering through some of the cytoplasm of the amoeba into the food vacuole. So on the left here we see a picture of a couple of food vacuoles, and what's happening in a food vacuole then is digestion. So digestive proteins are like molecular scissors, and they're going to create nutrient molecules from the prey. So here we have our little proteins chopping up the paramecium into bundles of nutrients. Let's just take protein for example. Let's say there's a bunch of paramecia proteins. Well, each of those paramecia proteins are composed of amino acids, but it's the same amino acids that the amoeba uses to build amoeba proteins. So what the amoeba is going to do is to break the chemical bonds between the amino acids. That's digestion. And so you're going to get a bundle of amino acids that the amoeba then can use to build amoeba proteins. We do the same kind of thing in our stomach. We have a special protein called pepsin, and it's a digestive protein that can break down the protein that we get in our diet. So when we break down the proteins in our diet, we are turning animal protein like a hamburger or plant protein into a bunch of amino acids, and then we are going to use those amino acids to build human proteins according to the recipes of our DNA. So back to the amoeba. Here we have a food vacuole. Digestion of the protein will occur here, and then the resulting amino acids will be transported through the membrane of the food vacuole into the cytoplasm. So the amoeba can use those amino acids to build amoeba proteins. In addition, of course, all the other nutrients found in a paramecium will be absorbed into the cytoplasm to sustain the chemistry of life. And recall, for bacteria, they are simply importing molecules from their environment, whereas larger cells, predatory cells like amoeba, are eating other living things and therefore have to do digestion. But then they import nutrient molecules into the cytoplasm. And so what happens now in the chemistry of life here is a lot like what's going on in a bacteria cell. Now, the plant cells are getting energy in an entirely different way. They're, of course, capturing sunlight energy to power this important chemistry going on in the chloroplasts. You'll recall in our abstract cell, cells have to get energy. Animal cells get energy by importing glucose. That's a molecule, so molecules can contain energy. and then they break down the glucose to extract that energy. But plant cells are getting energy in a different fashion. They're collecting, they're harnessing, they're absorbing light energy. And of course, this powers a very complicated chemical process called photosynthesis, where plant cells and other photosynthetic organisms are using carbon dioxide and water molecules to build sugar with some waste oxygen, and that's powered by light. And we've already learned that photosynthesis is occurring in the chloroplasts of the cells. Now, bacteria can also do photosynthesis. In fact, photosynthesis was invented by bacteria. So bacteria don't have chloroplasts. And in fact, bacteria, again, are almost the size of chloroplasts. Here's one type of bacteria, cyanobacteria, that is similar to some of the earliest forms of bacteria that did this modern form of photosynthesis. So if we think about our kind of three cells here, but two categories, the complex cells on the right and then the simple cell on the left, some bacteria perform photosynthesis. No animals or amoeba perform photosynthesis. And of course, the plants and creatures, single-cell creatures like Euglena, do perform photosynthesis. So photosynthesis is not a universal feature of cells. Again, some bacteria perform photosynthesis, but they do not have chloroplasts, whereas the complex cells, the large complex cells, perform photosynthesis because within those cells are these structures called chloroplasts. There's another difference between these cells, and that is that large complex cells all have mitochondria. In the case of animals and single-cell creatures like amoeba, and plants and single-celled organisms like Euglena, they possess mitochondria to power their life functions. Bacteria do not. In fact, the bacteria cell is about the size of mitochondria, so it would be impossible for bacteria cells to have mitochondria. And again, in another lesson, we'll examine why it is that mitochondria are the size of bacteria. And the answer will be that scientists believe that mitochondria today were once free-living bacteria. Another difference is the presence of a nucleus. Now, for large and complex cells, the DNA within the cell is surrounded by a membrane called the nuclear membrane. So animal cells, amoeba, plant cells, and Euglena all have a nucleus, a membrane surrounding their DNA. Bacteria do not have a membrane surrounding the DNA. All cells have DNA, but bacteria don't have that membrane, so they don't have a nucleus. So therefore, the nucleus, the presence of a nucleus, is not a universal feature of cells. Let's take a look at a couple of pictures of some organisms that have a nucleus. Here's a paramecium, and you can clearly see with this special lighting the nucleus of the paramecium. And of course, on the left here we have the nucleus of a plant cell, and on the right, the nuclei of some animal cells there. Another difference is chromosome structure. In the bacterium, the DNA exists as a circular molecule of DNA. In complex cells, the DNA exists as straight chromosomes. And these large complex cells may have many chromosomes within the nucleus of the cell. So for all complex cells, the DNA exists as straight chromosomes, whereas for bacteria, it's a circular chromosome. Now, in a previous lesson about DNA, we learned that bacteria duplicate their circular chromosome and then deliver identical copies to the two daughter cells, as you see in the pictures here. Well, complex cells do something of the similar kind of thing. They're going to duplicate their chromosomes and then deliver identical sets of chromosomes to two daughter cells. Let's take a look. All right, here we see a paramecium in the early stages of cell division. Now, by this time, it has duplicated its chromosomes, and the nuclear membrane has broken down so that the identical sets of chromosomes can be delivered to each side of the cell. And then the cell's going to divide right here. We're starting to see the groove here that will mark the separation between the two daughter cells. By the way, we see some bacteria in the environment. That would have been food that's powering this paramecium. So we'll go a little forward in time. and we have separation. Let's take a look at what amoeba would look like. All right, here we have one parental amoeba cell that is dividing into two. This has been speeded up. And the moment of separation. So we've got one amoeba dividing into two. Now DNA, we've learned, stores the recipes to build each kind of protein, and we called that recipe a gene. So here we have a cartoon cell up top here, an amoeba type cell making different kinds of proteins so it can do its thing. Well, each of these proteins has its recipe stored somewhere along the chromosomes of that cell, and a gene would be the recipe to make one particular kind of protein. Now, large and complex cells make more kinds of proteins than bacteria do. And large and complex cells have more DNA than bacteria to code for those extra proteins. Here's a fun fact. A single human cell's DNA is about 6.5 feet long. Lining up the DNA from all the cells in the body would stretch from Earth to the sun and back 60 times. And if you take all the DNA from all humans on planet Earth, if you line up that DNA, it would stretch to the Andromeda Galaxy and back nearly 2.5 times. Now, with lots of DNA, complex cells evolved packaging proteins called histones to coil up the DNA so it could be efficiently managed during cell division. So here's the DNA molecule on the right, and large complex cells have evolved a bunch of different kinds of proteins that allow the DNA to be wound up and coiled up so that the cell can better manage the larger amount of DNA that it requires to run a larger cell. Managing more DNA and making more proteins required more energy. And so now we get back to the importance of mitochondria. Mitochondria provided the energy for cells to get bigger. The presence of many mitochondria allowed cells to experiment with new and more numerous genes. Cells could now evolve to be larger, more active, and more complex with a greater range of capabilities. So we'll end then with some universal features of cells. Cells have a boundary that controls the entry of nutrients and the exit of waste, and that, of course, is the cell membrane. Cells have a fluid interior where the chemistry of life occurs, the cytoplasm. Cells extract energy and resources from the environment to maintain life. Cells have a team of molecular machines called proteins that do a wide variety of tasks needed to keep the cell alive. And recall, these are adaptations, these proteins. They're involved in processing energy, building the cell's molecular parts, repairing damage, carrying out the cell reproduction. And finally, cells have a set of information molecules called DNA that carry the instructions for making the proteins. DNA can duplicate, allowing for reproduction, and DNA can mutate, allowing for evolution. The non-universal features of cells would be mitochondria, only present in the large complex cells, which is pretty much everything on planet Earth except for bacteria type of cells. Not universal would be the nucleus, only found in large complex cells. Again, bacteria don't have them. And photosynthesis, only found in plants and some single-celled species, like Euglena or some bacteria. So this concludes our comparison of some of the different kinds of cells on planet Earth.